Technology. The topic of discussion today is very basic yet very complicated because if we are able to understand what a deribonucleic acid molecule is and how it works, we will be able to understand almost every concept in biology. So today we know that DNA is a genetic material. However, this result has been obtained after a lot of efforts, some of which we should cover today. So before it was identified that DNA is actually genetic material, there were certain criteria set up to test the available biomolecules uh, on uh, on this litmus, and then identify whether proteins or carbohydrates or lipids or nucleic acids are the genetic materials. So a genetic material must meet four criteria. Number one, it should be able to carry information. From information, we mean that it should be able to possess the information to form the proteins required for the daily life function and reproduction, etc., as well as the molecules that are not directly proteins, but they still they're functional inside the cell, for example, RNA molecules. Secondly, a genetic material should be able to be transmitted faithfully. This means that when a cell replicates to form daughter cells, or when an individual reproduces to form the offsprings, the genetic material should be transmitted in a way that it does not change the overall appearance to a significant degree, or it does not disturb the function, or it does not lead to speciation. Thirdly, a genetic material should be able to replicate. This means that it should be able to make an exact replica of itself without the introduction of changes. And number four, that a genetic material should be able to carry a certain degree of variation. This variation will lead to evolution, and this will lead to the individuals who are better adapted to the environment. So, a genetic material should not only be able to conserve its information, uh, at the same time, it should be able to introduce certain variations in it to lead or to make genetic diversity. So there have been many efforts starting from the Mendelian time after Mendel proposed his laws of segregation and independent assortment. However, these laws remain undiscovered until early 1900s, where two groups of scientists independently worked on Mendel's principles and identified that they were true. By early 1900s, it was known that the chromosomes are the entities that carry genetic information. However, as we know that chromosomes are made up of not only a nucleic acid named DNA, but also proteins. So, if we briefly revisit the timeline of genetics, we come to certain milestones. For example, it was Mendel who introduced the concept of heredity. Then it was Johnson who coined the term gene, because before this term, Mendel called these genes or the underlying things that control the traits as factors. Then came the famous animal geneticist Morgan, who worked on Drosophila extensively to not only rediscover Mendel's laws, but also he laid the foundation of genetics, and on his discoveries, even today's genetic principles are based. He discovered that genes are responsible for inheritance, and these genes they are present on chromosomes. However, it took almost 20 to 30 years to identify DNA as genetic material, and there were certain landmark experiments in elucidating this fact. The first landmark experiment was performed by Griffith, and they're famous as Griffith's transformation experiments. What he did, he used two strains of a disease-causing bacterium named Streptococcus pneumoniae that causes pneumonia. And this strain comes in two forms. One is virulent and one is avirulent. The virulent or the pathogenic strain is so because of the presence of a capsule, and therefore we call it a smooth strain because it produces smooth colonies on solid media. However, the avirulent or non-pathogenic strain lacks the capsule and it does not produce smooth, rather produces rough colonies, and this is the avalent strain. Similarly, they have two core types, 2 and 3, so there are four possible phenotypic combinations, either 2S or 3S or 2R or 4, the 3R. So the experiment consisted of a series of steps that were based on the conversion of smooth strain into rough strain and rough strain into smooth strain. However, the capsular trait was uh, uh, could be converted. However, the core types they were not changed. Uh, type 2 remained type 2, type 3 remained type 3. So this is a series of experiments that Griffith did. He inoculated the mice with living smooth bacteria, and mice died. And when he cultured the blood from dead mice, it resulted in the production of smooth colonies on nutrient media. When he inoculated the living mice with rough strain, the disease did not develop and the bacteria did not die. The mice did not die, and the body fluids did not resulted in any smooth bacterial colonies. However, when he mixed the living rough strains with heat-killed smooth strains and then injected the mice again developed the disease and died, and this resulted in the production of living smooth colonies. So here, Griffith concluded that something from the dead type 3S transformed type 2R into type 3S. This means that this is a reversible phenotype, and this process he called the transformation. And the thing that did this, he termed it as transforming principle. Then Leibniz, he actually works on the chemistry of deoxyribonucleic acid and discovered that it is made of four nucleotides. And these nucleotides consisted of phosphates, sugars, and four bases. Then this is a second landmark experiment which we shall discuss in detail afterwards. He showed that the transforming principle in Griffith's experiment was actually DNA. So this is the experiment which I was talking about. What he did that he tried to identify the exact actual biomolecule that did transformation. So inside cells, as it was known that the major components are nucleic acids consisting of two major types, DNA and RNA, then proteins, carbohydrates, and lipids. He made cell extracts from the smooth virulent cells, and each of them, of course, contained all of these macromolecules. But he tried to degrade each biomolecules separately by using specific degrading enzymes. He mixed each extract with type 2 rough cells to test for transformation, and the only extract that contained DNA, which was transformed into smooth strains. Whereas in the rest of the mixtures where he used um, uh, an RNase to degrade RNA did not result in smooth colony, did not change the phenotype, or a protease that degraded the proteins did not change the phenotype. However, the only change was visible when the DNA was present in the mixture. The chemical composition of DNA was elucidated by Shagaf. He showed that the proportion of A and G collectively is equal to the proportion of T and C, and both roughly equal 50%. He used this data to this data was ultimately used to identify the double helical nature of DNA by Watson and Crick. Um, This data was helpful in elucidating the distance between bases, the length of the helical turn, and the rise of the helix. In the meanwhile, when Watson and Crick were working on the elucidation of DNA structure, Franklin did the crystallographic studies, and she found that DNA is a double helical molecule, and she used both the dry and wet forms of DNA. This is the principle of X-ray crystallography, whereas the mixture of the suspension of the biomolecule is placed in the middle from which the X-rays pass, and they then get diffracted onto a film, and the image on the film is actually what the structure of the diffracting biomolecule is. The third landmark experiment in which settled the debate that whether DNA or protein is the genetic material was Hershey and Chase experiment, in which they used a bacteriophage, a virus that attacks bacterium. This is the life cycle of bacteriophage, where the virus attaches to the cell surface of the bacterium, injects its DNA, which then takes on the resources inside the cell. The virons or the phage particles, they are assembled, and the host cell is then lysed, and the new phages are released to continue the same cycle again. The important point in this was, as we now know, that the DNA is injected in the cell, but the viral particle consists of a protein coat and genetic material consisting of DNA within the coat. So Hershey and Chase aimed to identify whether it's the protein coat that enters the cell or it's the DNA that enters the cell. In order to distinguish these two, they used the radioisotopic forms of certain elements. For example, the proteins, they contain sulfur, which is absent in DNA, and DNA contains phosphorus, which is absent in proteins. So they labeled DNA with radioisotopic phosphorus, and they labeled protein with radioisotopic sulfur. And the principle was the rationale was, if protein coat enters the cell, then we should find the radioisotopic sulfur in the plaques. And if DNA enters the cell, then radioisotopic phosphorus should be present. So they infected the non-radioactive E. coli strains with radioactively labeled phages, and when the cells they lysed, what was present inside bacteria? Was it the phosphorus, isotopic phosphorus, or radioactive sulfur? So this is the experiment, and in which the one was the culture contained labeled phosphorus, and one culture contained labeled sulfur in the bacteriophages, and the bacteria, the hosts, they were unlabeled. So after the hosts, they were separated from bacterial cells, the viable phages that were produced contained radioactive phosphorus. However, from the unlabeled infected bacteria on the unlabeled phages they were produced. This showed that the radioactivity that was present inside this bacterial cells was from the nucleic acid. This means that viruses, they injected their DNA into the